All right. Well, I'd like to thank you for joining us for another of the CORES lectures. Today, we're lucky to have Mike Frank here to talk with us. Mike is a professor of psychology and the David and Lucile Packard Professor of Human Biology at Stanford and director of the Symbolic Systems program. His lab is interested in how children learn language and how this relates to their social interactions. And in recent years, his lab has taken a really data-intensive approach to asking these questions through large-scale studies, including Word Bank and MetaLab. He's also been involved in the movement to improve the reproducibility of psychological science for nearly a decade, including his participation in the original reproducibility project, Psychology, and more recently as one of the leaders of the Mini Babies Project, a collaborative project to replicate influential experiments in developmental psychology. He also teaches the highly regarded experimental methods course in the Stanford Psychology Department, which has introduced many of our graduate students to the fundamentals of doing open and reproducible science. So today he's going to talk with us about the concept of the reproducible manuscript. Thanks for joining us, Mike.
Hey, thanks so much for having me, Russ. Thanks for all the work that you and Maya are doing with CORES. So let me share my presentation today. This is really a pleasure to prepare this presentation, as this is a topic that I've worked on and thought about for quite a long time, but it's not something that I've had the opportunity to speak on in exactly this forum. So this should be a little bit of fun. So let me begin with a story. As Russ mentioned, I'm a developmental psychologist and I study infants and young children. I got my start working in an infancy lab, and this is really I don't know if it's my first paper submitted, but it was either first or second paper that I submitted with several of my mentors from even before I went to grad school. And one of the key results, maybe the key result in this paper, was that we had taught 5-month-olds something. We got them bored with a particular stimulus, and then they showed that they had learned some regularity because they then dishabituated. Their looking times went back up again when they saw a stimulus that differed from it on some abstract or formal dimension compared to a control. That's the result that's shown here. And this paper went through peer review. 2 expert reviewers and an expert editor looked it over. My co-authors and I looked it over through several revisions. And it was published in 2009. Around 2012 or so, I was involved in a debate around some theoretical issues. And the person I was debating with brought up this paper. So I started thinking that I would sort through the data from the paper to try to understand whether their point really applied. It was kind of a side point about analyzing these data. And I noticed something really weird. This graph, which is showing infants' looking times, was showing looking times below 5 seconds for the entire experiment. This seemed totally crazy to me. Really, really very strange because infants in this experiment would fixate for many, many seconds, sometimes 60 seconds or 30 seconds after a couple trials. It seemed very odd. Unfortunately, it was odd. There was an error in the analytic code for producing this figure. Luckily, it applied only to the training trials and not to the test trials here. So the inferential conclusions that I reported were valid. Nevertheless, I had to correct the article in print. Here's the corrected figure, which you can see has habituation curves that look much more normal, going from about 60 seconds down to 15, eventually down to 5 or 10. And then dishabituation looks like it should, which is a difference from that earlier baseline established during habituation. The axes of these graphs are fundamentally different. Despite that, my co-authors and the reviewers and editor, all of whom work in this domain, had failed to notice this error and had failed to correct it. Luckily, I still had some aspects of the code that generated the figure, and so it was pretty easy for me to find the issue. But the workflow that I used to support this figure was really fundamentally a bit inefficient. So there were some Excel data. I then wrote some MATLAB code to analyze those data. I exported the figure as an EPS file and tweaked it in Illustrator for appearance. Then I dragged it into Word and submitted it. So I was able to track the process and figure out that somewhere in the MATLAB code section, there was a problem. And then I looked at the code that I had written, which in hindsight doesn't look great, doesn't smell great, as folks say about this kind of code. And I noticed that deep in the specification of the analytic variables, I had these variables that were helpfully named HM7, HM8, and HM9, and so forth. Those are not the right numbers. And that took a substantial amount of anthropology of my own, or archaeology of my own code to figure out. So in this case, really, it took a lot of digging around to figure out where my error had come in. I felt very upset about this. I was glad that I found it myself rather than somebody else finding it, but it was still an upsetting moment. I remember exactly where I was. I was on a plane, actually, and my kind of blood ran cold, and I didn't know the scope of the error. I thought I was going to have to retract this manuscript. It was really a very bad feeling. And as I reflected on this later, I realized there were some real fundamental issues in my workflow that I had never really been trained to create a workflow that allowed me to document and reproduce the provenance of the numbers in the manuscript, and that then the coding practices that I were using really didn't create an ecosystem where it was obvious what was being done, where it was labeled in a way that was human readable, and where I could really clearly check my work, or another person for that matter could really check my work. Now, as I've gained more experience in this space, I realized that that was certainly true of my work, but it was not just me. Here's a small snippet of Stata code. This is from a paper that was published in Psychological Science. And in the class that I referred to, Psych 251, which is our introduction to laboratory methods for graduate students, we download papers, data, and code, and try to reproduce some of the analytic findings from those papers. More about that later in the talk. And this is a code snippet from one of those papers. Now, an intrepid grad student, Kensign Lewis, was working on this particular paper and was trying to reproduce this code by rewriting it into R. And we looked at this code and said, this is really very, very tough to understand. It seemed like there's some recoding happening. So there's a composite variable that's being made based on some non-human readable labels for particular questions. This is really tough stuff. And the code went on for maybe 2-3 pages worth of this kind of recoding. And it, between Kensign and me and Andrew Lampinen, one of the TAs in the course, we rewrote the code in R a couple of times, finally coming up with the conclusion that in fact, some of these statements were not only difficult to translate, they were in fact incorrect based on the description in the paper. Peterson, the original author here, took this very seriously, immediately went and corrected the analyses and issued a core addendum. So did exactly the right thing here. So this was an experience that suggested that, these problems of kind of issues of errors in code and in data, code interacting with data, really can have fairly pervasive effects, especially like in this example and in the one before, the code is not human readable. And these are best case scenarios, right? These are cases where we have the code, we have the data, and we're still not doing so well in terms of catching errors prior to publication. XKCD often says it best, right? This is the pro tip: never look in somebody else's documents folder. This is referring to kind of version control issues, but I think the issue is more general, that in the sciences, we don't tend to look at the source code or organization or file and data management infrastructure. I was certainly trained as a grad student that I developed all that stuff on my own. There wasn't a lab standard protocol. I didn't take coursework in this sort of thing, and so forth. And these kinds of issues of kind of errors within the chain of analysis can have really far-reaching effects. Just to take one example that was in the news, a very influential paper in economics by Reinhardt and Rogoff was used to argue for austerity policies, but famously the conclusions were invalidated because they were due to a miscoding error in some Excel formulas. So there are many, many of these sorts of errors that have been, some have been well publicized, others have been kept relatively under wraps, like the two I started with. But in general, this has led us to worry that things are not entirely fine. Now, in the face of a variety of anecdotes around problems in the scientific ecosystem, we might have a number of different responses. One might be to think that everything is on fire, and the other might be to carry on as normal. And it's hard to say which of those is really rational. In fact, for much of my scientific career, there's been a kind of continual discourse in social media and at our conferences and in the field of psychology more generally about what the right adaptive response is to the situation around reproducibility and replicability, in part because anecdotes plurally don't make data, and it's very hard to know how systematic and pervasive issues are. So what I want to talk about in the remainder of this talk is some attempts to understand the systematicity and pervasiveness of these issues, and maybe think about some solutions that are out there. So one way of describing the issue that I want to discuss is about thinking about a scientific paper as resulting from an analysis pipeline that starts with the digitization, the collection of raw data in some form, then their digitization and inclusion as computer files, their processing, pre-processing, filtering, munging, and so forth, their analysis via descriptive statistics, inference, visualization, and so forth, and their write-up into a manuscript and their dissemination as mostly text, slides, occasionally figures, and a research report. And if we think about the goal that we all have is to ensure the integrity of this chain such that the data that are collected are represented appropriately in the reports and dissemination that we give. So that's the broader goal. And the problem, the basic, is the world on fire or not problem is how do we understand the extent to which integrity is ensured in this chain? Do we see high profile mistakes as evidence that there's fundamental problems in the integrity here, or are they occasional one-off errors that are regrettable but are not indicative of the broader issues in the ecosystem? So that's where things stood for me for a little while, that I knew that I had made mistakes, that I saw that others had made mistakes, but I didn't really know what to do. And so for me, it was really transformative and interesting to get introduced via actually the Reproducibility Project in psychology originally, get introduced to what I now think of as the Metascience Policy approach, which is that we can treat issues in the scientific ecosystem as issues like any other policy ecosystem, where we can identify a problem, define and measure outcomes, do policy interventions and analyze the potential consequences of those policy interventions, and make recommendations for further policy at the level of universities, at the level of journals, at the level of funders, and so forth. That the scientific ecosystem can be subject to the same kind of measurement and policy intervention that social scientists do in other aspects of the social policy ecosystem. So for me, this felt very transformative, very exciting, because it opened up a whole new way of trying to understand the problems that we face and deal with them. So to illustrate this, I want to start with a well-studied example in the policy metascience ecosystem that you probably are familiar with. But excuse me, but it does nicely illustrate the basic issues that people have grappled with in this space. So some terminological definition here. There's a lot of confusion around terms like reproducibility and replicability. I use these terms, and I think there's an emerging consensus around these terms that if you have the same code and the same data and you produce something like the same result, that result is termed reproducible or computationally or analytically reproducible. If it's the same code or same analysis, but on different data, that's termed replicable. That's a replication that's happening. If you're using different code or analysis specifications on the same data, that's a robustness check, or a sensitivity check in some people's terms. And different analyses and different data sets, you might call that a generalizable finding, although that term has a broader set of meanings. Of course, all of these are critical outcomes that we want to encourage in our scientific ecosystem, but we can focus on individual parts of the ecosystem and individual outcomes that we care about the most. And so replicability has been a key target, especially in psychology, where it's relatively cheap and relatively easy to gather new data sets and subject them to the same analysis and try to understand if the results are similar. So most folks here are probably familiar with the Open Science Framework 2015 paper. I was really proud to be a part of this collaboration to replicate 100 studies from psychology journals in 2008. 4 of these dots on the graph, each representing a study, came from the initial iteration of my class. And the goal of the study was to measure replicability. The title of the paper is Estimating the Reproducibility of Psychological Science. And there's several things I might correct about that title. It's not reproducibility, it's replicability. And it's not of all of psychological science, it's of a particular sample. So it's more like measuring reproducibility in a particular sample of papers from psychology. So Folks are familiar probably with the disappointing result of this study, which is only about a third of the projects worked by the various definitions of replication that were used in this project, a statistically significant finding, a subjective impression of replication success, and so forth. But there was also a tremendous amount of ink spilled around this paper in that it had some major limitations that I've mentioned. The sampling scheme was very much non-random, non-representative. So the estimate does not generalize to all of psychology. The power on some of these replications was relatively limited. So the replications may have been underpowered to detect smaller effects than the original. And there were major questions raised about some procedural fidelity choices, so some of which were later chased down and shown to be reasonable. But these were questions that were raised at the time. Now, critically, because this is a meta-scientific policy ecosystem, and this is a scientific study about a particular measurement in that ecosystem, some of the limitations can be addressed by further studies. And this was what was inspirational to me, is that although this study was important, it was not the endpoint, but rather the starting point. And so you can see further studies, and now there's quite a voluminous literature about this that address these issues. Just to cite one of these, this is a paper by Kammerer 2018, so about 3 years later, that addressed the limitations of the earlier study regarding sampling power and fidelity by doing high fidelity, very high power replications of key findings from a systematic sample from the journals Nature and Science, in which the entire sample actually was replicated. So all 20 of the findings that met the inclusion criterion were replicated. So this estimate now does generalize to that particular journal set pretty well. You could ask about temporal trends. The power was very high in the initial replications, and then if the replication failed, they actually did a second round with even higher power. So that was very impressive. And they gathered input from some of the original authors pretty extensively to try to correct some of the issues in replication fidelity. Whether they did so perfectly, I don't know, but I think they were much, much closer than in the replication project in psychology. So that all suggests that our estimates of our policy targets, like replicability, can be moving estimates that are estimated through better and better study designs. And I thought that was quite inspiring. Further, you're probably familiar with the broad universe of tools and policy interventions that have been intended to improve replicability. Pre-registration registries, badges, registered reports, transparency openness guidelines, a variety of these that all had aims around improving replicability as well as reproducibility. And now in this policy ecosystem, we can think about evaluating these as to whether they improve the outcomes of interest or whether they have other subsidiary benefits and whether those benefits are worth their costs. So that I find to be a really exciting and inspiring idea, that we can really do policy cost benefit analysis on these kinds of tweaks, nudges, and stronger regulatory approaches to science policy. Okay, how can we then use this kind of approach for the question of reproducibility? Well, let's start by breaking down reproducibility into two components. First, the availability of code and data for external reproducibility attempts. So if I want to understand whether your project is reproducible, I need to be able to get your data and probably also your code in order to do that reproducibility check. So if I suspect that there's some problem in the integrity of your analytic chain, I can't even test that for the most part without access, without transparency. But the second part is we also want to check whether that analytic chain has some integrity to it. So in some sense, the transparency, the availability of the materials is a first step only. And the second step is really checking the integrity of that chain. So the first thing I want to mention here is that the low availability of data and code renders most papers non-reproducible by external folks. This slide really draws on the research of my co-author and collaborator, Tom Hardwick, who's a co-author on this talk and has done a lot of the empirical work, has led a lot of the empirical work that I'm talking about here. So here's some data and code availability prevalence estimates from a variety of different subdisciplines. In psychology, 2014 to 2017, you have 2% data availability and 1% code availability. This is from a sampling procedure. So you really get a sense that in the broader field of psychology, essentially no one is sharing their code and data, even if they do in restricted subfields where norms have started to shift. Only doing a little bit better in social sciences and a bit better on data in biomedicine, but worse on code. Further, for many years, the standard for data sharing has been data available on request, but data tend not to be available from authors on request. One systematic sample from 2004 found that 27% of authors shared data according to requests. And that's generally true for other fields. There's an ecology survey with a similar estimate, and a very highly cited set of articles for which the data would be of very high value for reuse and reanalysis, found a data sharing rate of only 14%. This was a systematic study that Tom Hardwick and John Ioannidis did, where they reached out to try to recover those data and archive them for posterity. And they found that in many cases they had been lost, or simply the email addresses never responded. Okay, so from the external perspective, reproducibility is not there simply because of the lack of transparency. That is, the analytic chain may have been uninterrupted and the data may have been represented appropriately in these papers. That's entirely possible, at least for many of them, it's probably true, but we can't check that. And somebody who's skeptical doesn't have any real recourse in these cases because the data won't even be available on request. So that's unfortunate. And suggests that policies that encourage transparency are a critical part of ensuring analytic reproducibility. Now let's move on to think about the analytic pipeline and whether it has the kind of integrity we want in the general case. And this is important, right? Because if things are fine, if we basically have high integrity pipelines, then maybe the availability of code and data for checking is slightly less useful, is slightly less important. Now, of course, it's important for other reasons. Those data and code could be reused by creative scientists in a variety of ways that could speed up discovery. But maybe it's not a critical flaw in the research ecosystem because mostly the reports are correct in terms of their details. So how do we measure the integrity of the chain without the availability of code and data? Let me tell you about a clever way that one research group has worked on this. This is originally work by Baker and Weikertz. So Baker and Weikertz made an observation about the psychology literature, which is that the American Psychological Association mandates a particular statistical format for reporting. Like in this case, T of 46 degrees of freedom is 1.28, P equals 0.207. So this reporting format is redundant because any two of these numbers can be used to compute the third. The p-value is redundant with the test statistic and the degrees of freedom and so on. That provides an opportunity for checking aspects of the reporting chain. Because if these three numbers don't match up with one another, we know the chain was broken somewhere. Because it's impossible mathematically for all three numbers not to match up unless there's been some typo or issue somewhere in this analytic chain leading to the report. Now, of course, if the numbers match, that doesn't mean that they're the right numbers. It doesn't mean that the right data were analyzed and so forth. So this is a lower bound on cases where there's been some kind of error in reporting, but it provides an innovative method for doing this. And so here's the result here. So the key place to look is the total number of statistics examined here is 600 odd p less than 0.05 statistics, and there are 122 errors, 18%, with some of these being what they called gross errors, meaning errors that changed whether the p-value fell above or below the alpha threshold of 0.05. So that might change the inferential conclusions of the test. And there were more such gross errors and more errors overall when the p-values were below 0.05, suggesting that there might be some mechanisms at work related specifically to the, quote, significant results in these findings, in these papers. So this is from a sample of hand-coded manuscripts that these researchers examined. But in fact, because of this reporting requirement is so strict, automated methods become available. And so this is a very clever application of automated methods by Michelle Nuiten and her colleagues. that looked at the proportion of tests across all APA journals, across thousands of papers. This is the proportion of inconsistent tests per article. So about 10% of all tests reported in articles had some inconsistencies, and maybe 1 or 2% of those had gross inconsistencies. Here's this same data presented in a slightly different way. This is the proportion of articles with inconsistencies, or percentage here. So about 50% of all psych articles had at least one statistical inconsistency, suggesting that there's some real potential problems in this chain from data to reporting, possibly here maybe closer to the reporting end, having to do with cut and paste errors, having to do with rounding errors, having to do with general biases in reporting, misreporting statistical values here. But this for me was very concerning because it suggested general problems in the analytic chain that were widespread and systematic. And really, I think whatever our scientific values are, we can all agree that it doesn't make any sense to have scientific papers with statistical tests that just are literally impossible, right? So correcting this kind of issue is just, it seems like a key problem. And if they're as widespread as 10%, it seems like a very important issue to address. So that was my context. And then thinking with a Metascience Policy hat on, I got a very interesting email in about 2015 from a guy named Steve Sloman, who was taking over as an editor at the journal Cognition, which is a major disciplinary journal for cognitive science, child language, psycholinguistics, and so forth, my subfields. And Tom was just joining my lab as a postdoc. And Steve Sloman said, hey, I'm taking over as editor-in-chief of Cognition. And, you know, I know you all are talking about all these guidelines and so forth, but I'm editor and I have absolute power. And I think data sharing is the right thing to do. You have to share your data if you publish at Cognition. And I was very inspired by this. Actually, I thought it was really a brave move and was really, I admired him because he had taken over this journal on the condition that he'd be allowed to put this policy into place. I thought that was very exciting. And Tom did too. And he pointed out that we could do a policy study of this intervention. So I became an associate editor at the journal and edited papers there for a number of years. And at the same time, Tom and I did a study of analytic reproducibility at Cognition. Now, let me say here that this effort was led by Tom, but a wide variety of folks took part in this study, including Maya and many other folks, some of whom were at Stanford at the time, others of whom heard the bat signal and came and joined in and took part in our attempts to code articles and to do analytic reproducibility attempts. So this was a ton of work. Tom really did a huge amount of work coordinating this project. And the result is a case study of analytic reproducibility in psychology around a policy intervention. So Tom started to look at the literature on policy interventions to try to understand ways that we might really get a sense of the effect of the policy on the behavior. Now, we didn't pursue really strong causal inference designs, in part because of the immense cost of coding articles. So coding whether an article is reproducible is really very difficult. It takes a lot of time and a lot of effort. So doing a difference design might have been too tough, but we did do a regression discontinuity design. So the first aim of the project was to look at how in principle reproducible the results were from these papers. So we took a random sample of papers from before the policy, about 400 of those, and then all 200 or so of the ones that were published after the policy. And we looked at their data availability, whether the data were complete and reusable, and whether they were well enough documented to be in principle used again. What we found was pre-policy, there was about 22% reusability. Post-policy, that went up to 63%. The regression discontinuity was pretty clear here. There was also a bit of a delay in policy adoption, so compliance wasn't perfect despite the mandate, despite the system requiring upload of data and so forth. Still, a number of papers the associate editors just didn't check. The data never got uploaded and so forth. So the policy did boost the outcome, and it seemed to do so more than for comparable journals in a kind of informal analysis that we did. But it really, some kind of compliance checking was necessary here. So That's initial positive evidence about transparency step here, that a policy encouraging transparency might in principle raise the possibility of reproducibility. But we wanted to ask for those papers, those 63% for which we had data, how reproducible were they in fact? And this is, I think, what really is novel about the study, in the sense that we were really trying to check the analytic chain and essentially every step of the way from data to report, which we could only do because of the success of the transparency policy. So what we did was identify a straightforward and substantive finding from a pseudo-random subset of 35 articles. So these were articles where we had, in principle, reusable data, and we identified an outcome that we were going to try to reproduce, a numerical outcome or set of outcomes. These had to be straightforward enough that they were covered in an introductory stats textbook. So an undergraduate level textbook, ANOVA, t-test, linear model, and so forth. Couldn't be a complex model or a reinforcement learning algorithm or something like that. And we did this by co-piloting. So an initial pilot tried to reproduce this analysis, writing code to find the same numbers from the author's data as the authors reported in their paper. And then we pulled in a co-pilot in every case who checked that and ensured that they believed the reproduction attempt had done everything possible to try to find the numbers, or that they were correctly finding the numbers in the case that it was successful. And we reached out to the authors in every case where we were unsuccessful, and requested clarification. And we had, in this study, actually, I think 100% author response rate. So every author team got back to us and tried to help us clarify the issues. We coded as a major error those cases where there was a greater than 10% discrepancy between the reported value and what we got. And so an article was then coded as not fully reproducible if it had one or more major errors. Okay, so here are all of our 35 articles. 11 of those 35 were fully reproducible, meaning the pilot and co-pilot could get all of the numbers in the selected section without any help from the author. Another 11 were fully reproducible once the authors got back to us. And then 13 of those 35 were not fully reproducible, even in the case that the authors got back to us and shared the information they had. You could see there, these are the statistics that we had. So n's, confidence intervals, means, degrees of freedom, and so forth. And you can see the kind of approximate magnitude of the mismatches here. You can see that they're spread throughout the set of statistics that we had, including simply the number of participants in the study, for example. We checked about 1,300 values with 64 major numerical errors, about 5% rate. Critically, there was no case where the reproducibility issue appeared to seriously undermine the inferential conclusions, although in 3 cases that wasn't clear because we really couldn't figure out exactly what the right result should have been. Several authors, as I believe 3 authors in the study, expressed an intention to submit a correction. I'm not sure if all of those followed up, but at least 3 felt that this was significant enough that they should report it to the editorial team. So that's a 10% correction rate for this study. And also importantly, this is not sustainable. You can't really do this other than as an exercise. Some of these reproducibility checks took up to 25 hours. None of them took less than 2 hours. They're like doing very hard crossword puzzles. So what went wrong? For those papers that were not reproducible without the author as assistance, there were some analytic issues, some resolved, some unresolved. There were some typos, quite a number of typos. We couldn't track down why some issues cropped up. Several cases, the data that they had uploaded were incorrect, or they simply didn't have the correct data anymore. And often the analysis was not specified concretely enough to allow us to, and in some cases them, to recover exactly the numbers that they had reported. In the cases that were resolved, usually there were details that were missing from the analysis that once specified would allow us to recover the correct numbers. Occasionally there were data files as well, or sometimes pieces of data like which participants exactly were excluded. Okay, so as I said, there was no case where the reproducibility issue clearly undermined the substantive conclusion. There certainly are cases like that, and we've experienced them in other reproducibility audit studies, often less systematically conducted because we're interested in a particular finding or whatever. But these were like putting together a flat pack furniture with a really terrible instruction manual. It was very, very hard work. And it suggested that the data alone is not sufficient for ensuring analytic reproducibility in any kind of scalable or kind of high coverage way. Instead, it's really critical to have, at a minimum, code shared along with data in order to facilitate a clear specification of exactly what's being done. Because Tom is a glutton for punishment and believes in replication, we actually replicated this study with another sample of manuscripts from the journal Psychological Science. There's an additional sample of 25 manuscripts. The results were very similar. Our protocol differed very slightly, but the meta-analytic means stayed very close to about 1/3 being reproducible before author contact and 2/3 being reproducible after author contact. Okay, so two components of reproducibility, right? Availability of code and data for external reproducibility. Seems clear based on the code requests, their data request studies and general surveys of the literature that data are not available, neither are code. And our study suggests further that code and data are required for reproducibility externally to be at all scalable. So transparency policies thus must require code and data. And with respect to the integrity of the analytic chain for internal and external reproducibility, it seems clear that this chain is broken fairly consistently, both by virtue of the typo studies and by virtue of the broad taxonomy of errors that we identified with high prevalence in our study. So that suggests that we need a revision to our workflow around scientific reporting. What does that revision look like? I would suggest that it's the adoption of reproducible reports, reproducible manuscripts. So it's likely that in this lecture series, you've already heard this wonderful quote from Buckheit and Donahoe, their provocative statement that a scientific publication is not the scholarship itself, it is merely advertising of the scholarship. The actual scholarship is the complete software development environment and the complete set of instructions which generated the figures. So the idea of a reproducible report is that that complete environment and set of instructions is in fact the report itself. So the report itself is a piece of code that when combined with data will create the disseminated document. So a reproducible report is a document for which the entire analysis pipeline from primary data all the way to reported numerical values can be recreated by an independent scientist. So that's the proposed policy intervention here, is that we teach and promote and maybe in some cases even require a reproducible report workflow. Why write reproducible reports? Well, I've been arguing from a couple of different grounds here that I'll try to take apart here. The first is that provenance, transparency, and verification is very important. And so in order to know what the numbers in a particular manuscript mean, we need to know where they're from. And so this kind of transparent sharing of a reproducible manuscript enables independent verification of where those numbers come from. The corresponding side to that is that error detection and mitigation becomes possible. As I started with, it was clear that disorganized workflows with many manual tasks create opportunities for error. In a reproducible report, the flow of information is documented by code and directed by code, and it's much easier to audit. Further, manual tasks like the ones that the copy and pastes and hand rounding of statistics get avoided, reducing opportunities for the kind of typos that show up in the statistical consistency tests that I mentioned. So the suggestion is that in reproducible manuscripts, we should see violations of statistical reporting possibility go down by a substantial fraction. Finally, and this is what I tell people when I teach my course, I do reproducible manuscripts because they are a good way to get things right, but they're also way easier and faster, actually. And so this is why I've become evangelical about this general framework. It's because we often need to rerun and reuse our analyses. We often have to reformat our papers. We often have to repurpose our code to make not just figures for a paper, but slides or posters. And in a reproducible workflow, it's way easier to build on that work, to reuse your code, to remember what you did previously. So I'm basically moving towards wrapping up because I've become fairly evangelical about reproducible manuscripts in general as a consequence of conducting these studies with Tom and our colleagues. And so I now go around giving talks on a particular reproducible paper ecosystem, which is R Markdown. It's not the only one, but it's one that has worked very well for me, and most of the papers coming out of my lab are now written in it. So we developed some tutorial materials to teach people how to use R Markdown for writing scientific papers. It turns out to be extremely easy. So in an hour or 90 minutes, you can learn most of what you need to write a paper by integrating code and prose and get it formatted up in a journal's requirements. I'll end by saying that our in-progress project is to write a broader book called Experimentology, an open science approach to experimental psychology methods. We're moving towards trying to get a shareable first draft of this book, which I'm using for my Psych 251 course now. The goal of this book is to really provide an introduction to the kinds of workflows that I think are critical for a working scientist to try to avoid the errors that I've documented here. So everybody makes errors. I make errors. I actually make probably more errors than most people because I'm by nature a sloppy person who tends to work very fast. And so in order to avoid errors in science, it's critical to adopt a workflow that allows for transparency and reproducibility from the ground up. That's the best way to encourage verification, encourage checking, and create workflows that minimize error and maximize the chance of reproducibility. Okay. Thanks very much. I'd love to engage with everybody around questions and hear your thoughts on this topic.
All right. Thanks very much, Mike. That was an awesome talk. So we can take questions through the Q&A. So if you're in the audience and would like to ask something. So the first question from Joyce, what are your thoughts on the upcoming NIH policy on data management and sharing? Do you have anything to say about that?
Yeah, thanks for the question, Joyce. That's really interesting. You know, I haven't reviewed this in depth. So one of my questions is going to be, does it allow for code sharing? That's kind of, or, you know, does it mandate code sharing? And my worry is that it doesn't, that code hasn't been in you know, some of the previous versions of this. So yeah, I guess I don't know. I haven't reviewed the final version of this, but that's one thing that I've found and that I hope I convinced you of in this presentation is that code sharing is really a critical part of reusing data. If you don't have code, you probably can't get the same numbers. By probably, I mean less than half of the time. So the other thing that I'd worry about, and that I worry about in another, with my other hat on, as a domain scientist, I'm really interested in gathering and aggregating data sets. And so data availability is really great, and funder mandates for data availability are critical. But it's also really critical that they be accessible and licensed for broad reuse. So my experience, I'm going to say bad things here, so apologies, but my experience with NIH data sharing mandates has been abysmal. I've downloaded a data, so I run an archive of children's vocabulary outcomes using a particular instrument, doesn't really matter the details. In a large NIH-funded, taxpayer-funded study that used that outcome measure with children on the autism spectrum, I was very interested in analyzing those anonymized, de-identified data that are not human subjects data, right? So these should be publicly shareable. They are indeed publicly shared on the NIH data archive. My process for recovering those data was to ask the help desk 3 or 4 times to figure out how to log into the site. Once I finally figured out how to log into the site, I realized that in order to access the data, I needed a signed institutional agreement. I had to find like the president of Stanford to sign the institutional agreement saying that I was allowed to access these taxpayer-funded non-human subjects data. We finally were able to put them in our data cart and download them about 4 or 5 months after we began this process. But we were resoundingly barred from any further sharing because they can only be shared through this format. So making them publicly available through the actual domain-specific data archive that we curate on this particular kind of data is, of course, impossible. So data access is not, data sharing, data management are great. Data access is the last step in the chain, and that's been where federal funder mandates have fallen short.
I'll just add that we have a talk coming up in a couple of weeks, I think, by John Borghi about sharing research objects that I think will touch on these issues about licensing and accessibility. I think that's exactly right. So, so another question from KN Yang. Thank you very much for the talk. It's very informative. Could you please share some strategies or tools that can be used to improve reproducibility of the research? Because a lot of times I need to do analysis back and forth. It's hard to check changes and versions of the data and results, particularly when the data size is big.
Great, Kay, and thanks. Yeah, that's always the challenge, is how to manage the analytic ecosystem in a way that ensures reproducibility. So a couple of points here. The first is that starting with version control is very important here. Version control provides a suite of tools for project management around branches in code, around tracking issues in code, around rolling back and naming versions and so forth. So if you want to try a lot of different things, it can be very useful to use a platform like Git and GitHub to manage the different branches that you're exploring. The second point I want to make is that factoring your analytic pipeline is very important, especially when you're dealing with very large data sets. So making sure that you separate pre-processing steps from munging steps, from inference and visualization steps, so that you can then archive intermediate files in more manageable formats. And so that you can then functionalize your exploration of different pathways. So for example, if you want to try 2 or 3 different data preps or data pre-processing choices and explore their final result in terms of your visualization and analysis, you want to factor those 2 tasks out, store intermediates, and create sets of intermediates to be explored, and then be able to do that functionally and compare them side by side. Rather than the alternative that I used to do, and that sometimes I still do if I'm going fast and then hate myself for later, is copy and paste the same pre-processing code 3 times, change the constants in it, and then pull up all 3 figures and try to page back and forth between them. That's not the way to do things. What you want to do is create an ecosystem where parameters control the pre-processing script. They're cached as clearly named intermediates, and then those intermediates can be compared to one another in a computationally manageable way in the last stage, the inferential and visualization stage of the pipeline.
That's great, thanks. So from Joyce Wang, sounds like there's a lot of room for improvement. Are there field standards for data formats? For example, when you download data from a paper, is it usually like an unorganized Dropbox, or are there agreed upon standards for file formats, the organization of folders?
Joyce, thanks for your good questions here. So in our cognition study, actually, Cognition, the journal, just assumed that what data are is an Excel file. And so they made room on the journal submission site for the upload of an Excel file. And that was it. In psychology, more generally, there is no one agreed on standard for what data look like by any stretch of the imagination. On the other hand, more broadly speaking, when you start to get into the statistics, data science, and library science, information science communities, there is a lot of really good guidance for how to organize data. Just to kind of plug a couple of things here, Broman and Wu have a beautiful paper about how to organize data in spreadsheets for computational reproducibility and clarity. So making rectangular data that are easily machine readable and have clear names. There's also a lot of really good documentation out there about how to create metadata that allows others to reuse your data. So in general, any kind of clearly documented and consistent format can be dealt with. The issues that are inconsistency and lack of documentation.
I'll just add, I put a couple of links in the chat. One is to a project that's still sort of ongoing that is called PsycDS, that's meant to basically provide a kind of a data organization standard for psychological research data. The other thing that I'm pointing to is something that we've been involved in creating called BIDS, the brain imaging data structure, which is basically a format or an organization standard and a metadata standard for brain imaging data that is now been, it actually has an extension process, so anybody can come along and sort of propose an extension, and it's been pretty broadly extended to many different data types. So those are a couple of examples to look at that are sort of related. All right. Adrian Muir says that 100%, original 100% author response rate in your study is fantastic, super heartening. Did you get 100% in your replication study too?
We didn't get 100%, but it was pretty close. The samples there are quite small. Let me emphasize a bunch of things that I think are unusual and not really generalizable about that high response rate. So the first is that I was an associate editor at the study and am generally at a well-known private institution in the United States. So the response rate on emails that I and my trainees send is probably higher than somebody at another institution or without a connection to the journal or community. So that's a first thing that kind of makes it unrepresentative, unfortunately. The second thing is that we had, before reaching out for data, put in between 5 and 20 hours probably worth of work. And so we were writing saying, hi, here's the project we're doing, here's the protocol. We have already worked extensively on your data set. Here's a report of what we've done and exactly where we ran into trouble. Can you clarify these specific issues? So there was a very well-documented and specific ask in our email, and we tried to word this in as constructive and positive a way as we possibly could. And so that, I believe, also led to a much higher response rate. In practice, the numbers for author contact are much closer to the ones that I mentioned in the audit studies earlier. So they're much closer to 15 to 25% of data sharing according to author contact. And that's probably because the audit studies are not conducted by individuals in the specific subfield, and also because the data request comes prior to any work having been done. So it's, hey, can you put in the effort? And one third factor is that these folks had already, in principle, deposited their data, so they'd done some kind of preparatory work. Often, when we reach out for data under other contexts, the authors say, oh, we'd love to share that, but we haven't prepared it for sharing. That might be weeks or months of work. We can't do that. And that's when we get a response. Sometimes people get that and think, oh God, and I've been overwhelmed by this sort of thing too. It's going to take a lot of work. Maybe I just won't write back at all. So I think our results there are heartening, but I do think, unfortunately, they're unrepresentative of scientists in the wild interacting. There are a bunch of factors that make them non-generalizable, even though I was very pleased that people wrote back to us. I don't think that happens generally.
So while we're waiting to see if anybody else has a question, I will ask one, which has to do with sort of specifying of versions of packages, right? I had an example where I'd shared data and code and somebody came back and they said, I can't reproduce your result. And it turned out like the newest version of a particular package gave you a different result.
Yeah, I mean, so this is certainly a substantial issue. Just to take an example, much of my subfield relies on a particular package for fitting mixed effects models, LME4. So the LME4 package has undergone substantial revisions in what kinds of models it can fit and will fit when it throws error messages and what sorts of fits it returns with or without errors. Those revisions have led to pretty substantial changes in the inferential conclusions of some papers. And sometimes there's essentially no way without package archaeology to recover which version was being used, right? So an example that I have used in my course of a really beautiful study on cross-cultural detection or cross-cultural reasoning about fairness, something we analyze every year in my course. There's this open data set. It's lovely. It shows that some countries see greater developmental trends in kids' appreciation of fairness and unfairness when it benefits them and so forth. We can't reproduce any of the analytic conclusions of the paper, even though all the numerical conclusions are perfectly good, because their random effect structure simply doesn't converge under the 3 or 4 versions of LME4 that we tried. And so the p-values are just totally different when we try different random effect structures. Okay, so that's the problem. It is a really big problem. Why am I not talking about it here? Well, mostly because I think it's dispiriting to people to see how hard it is to make like just a basic mixed effects model completely reproducible. And so I've tried for the like 80-20 solution. Often when I talk to psych audiences, I say, hey, give me your code, give me your data. Yeah, I know the numbers are going to vary, but that's going to be a lot of the puzzle here. And if you do it in a reproducible manuscript, we're going to know where the numbers came from, and we'll know that there's a package version problem if the numbers are different. In computationally intensive applications, that's probably not enough. And so the solution around containerizing and package management is probably going to be pretty good. So Docker is clearly the industry standard for this. Here's the problem for me is, package management should take care of this, and it doesn't in the R ecosystem. So Renv and before that Packard, I've found virtually unusable tools for package management, and I don't want to Dockerize the t-tests and ANOVAs in our simple papers. So I haven't gone to that step for a lot of papers, and I haven't taught people about it just because it feels like a lot more weight for that extra 20%. But in part, that's just because of where I am in terms of the relative computation load and so forth. Anyway, right. Long answer, but it's a very valid question, and probably it's the end game of reproducibility.
Yeah, just to mention, I forget the name of it. There's a package from Microsoft that will let you basically install all of the packages for R that were current as of a particular date. And that's seems to be a little bit easier than having to do the RN thing, like storing all the particular packages. But I agree, it's definitely much easier in the Python ecosystem than in the R ecosystem. And we've moved to using Docker as the solution for those sorts of things, even though it's not perfect, but I think it's a decent solution. Okay, if there's no other questions, I think we'll go ahead and thank Mike again for a really awesome talk, and we will see you all next week for Shilan, who's going to be talking about sort of a more hands-on approach to addressing these questions. So thank you all.
Thanks so much. Thanks for having me. Great to see you.
Bye-bye.
Bye.